Right, good morning everybody. Today we're going to be talking about oxidative metabolism, and specifically oxidative metabolism in the mitochondria. When we talk about oxidative metabolism, what is oxidation? What does it mean, oxidative? Does anyone know?
Loss of electrons.
Say again? Loss of electrons. Okay. Does everybody know this? Yeah. Okay. So oxidation is a loss of electrons. There are many different definitions. There are many different descriptions of what oxidation is. You know, it could be dehydrogenation or whatever. But the most important definition, the most important description is that it's losing of electrons. Okay. What's the opposite of oxidation? Reduction, and reduction is gaining of electrons. Okay, this is a fundamental thing that we're going to build on today in the lecture. In our metabolism, in our cell's metabolism, oxidation is one of the most important processes that happen, and they mostly happen in catabolism. What is catabolism? In catabolic pathways? Yeah, anybody? That's right. In breaking down nutrients, mostly nutrients, in order to transform the energy, the potential energy, the chemical energy that's stored in those nutrients, to transform it into other kinds of energy that we can then use to do some work, some useful work. Okay? So today we're going to look at how these oxidative pathways in general, how they work, and then we're going to look at a very specific, very important pathway, which is called the respiratory chain, or the electron transport chain, which is in mitochondria. We're going to look at in detail how these things work. So today's lecture is going to be mostly about electron transport, moving electrons from one thing to the other, which is the fundamental principle of any redox reaction. Redox? Yeah? Redox, reduction, oxidation, redox. Right. To start with, we're going to take an example of oxidation of glucose. Okay? Glucose, an important nutrient, important part of our catabolic pathways, breaking down of glucose. So we're going to look at glucose, but what we're going to show, looking at the metabolism of glucose, is true for many other substrates as well. For example, fatty acids, some amino acids, etc. All right. So this is the reaction of glucose with oxygen. Okay, the direct reaction of glucose with oxygen. How do we get this reaction to occur? Well, we take a little bit of glucose and we set it alight with a match or something. Okay, so this is the description of the burning of glucose. Okay, if we just burn it in our atmosphere, this is what's going to happen. Glucose is going to react with oxygen, and the products are carbon dioxide and water. What else is produced in this reaction? Yep.
Energy.
Is it really? Okay, energy is never produced. Okay, heat, very good. So heat is given off, correct? And light. Okay, presumably there's some flame. So light and heat there's energy being released, being transformed, being put to the surroundings, okay, as heat and light, correct. So we can put that there's some energy being transferred. Good. Is this a redox reaction? Is there any transfer of electrons from something to something else?
Yeah.
Okay. Well, where are the electrons transported? Where from, where to? We're burning glucose.
Yeah, but this is a long process until you get...
No, we're just literally burning glucose. Okay?
No idea.
Where are the electrons going from and to?
Oxygen is reduced and carbon is oxidized.
Okay, so there's a suggestion that carbon is oxidized and oxygen is reduced. Are there other suggestions?
The opposite.
The opposite?
Yeah.
Okay, so oxygen is oxidized and carbon is reduced. Anything else? Any other possibilities? There are some other elements there. Okay, how can you find out? How do you find out? Oxidation numbers. Okay, what are oxidation numbers? Yep.
I don't know. Never.
Can somebody explain what oxidation numbers are? Yeah. Okay, so those are formal charges that would appear on an atom in a substance if it basically took all the electrons that mostly spend the time around that atom. It's just a formal thing, okay, but it's actually useful for some calculations. For example, figuring out what is actually being reduced or oxidized here. Okay, what are the oxidation numbers here? You can just start with the ones that are clear to you, and then we can get to the other ones. Huh?
O2 is 0.
Very good. Okay, it's a pure substance. 0.
Carbon is 0 on the glucose.
No.
How do you know that?
It's H12 is 12. Plus 12.
Well, you don't usually multiply. Okay, so it's plus 1.
Plus 1.
Yeah.
O6 is minus 2. And then this is 1.
What is going to be the oxidation number here?
3, 6, 1, 0, 4.
Okay, so we've already had about all the numbers, all the possible numbers. Okay, we had 0, 4, 6.
It's 4.
Is it 4?
Yeah, because no, actually, yeah. No, it's 2. It's 2 because
Okay, so we had 2, 4, 6, 0.
no, it's actually 0.
Okay, can you explain why it's 0? Yep.
Me?
Yeah.
Oh, okay, you have 12 hydrogens,
Yeah.
and then you have minus 2 on oxygen, and minus 2 times 6 is minus 12.
Yep.
That means the carbon has to be 0.
Okay, 12 minus 12 has to be 0. Okay, I know it looks strange because it's not pure carbon, but formally, carbon in glucose or saccharide that looks like this, or carbohydrate that looks like this, is going to have the oxidation of 0. Okay, again, it's a formal thing of accounting for electrons. Okay, there's nothing weird about this carbon. Okay, it's actually bound to other things, but if you do the calculation, it looks like it's 0. Okay, so we have 0 here. What happens in the on the product side of the reaction?
Carbon
Okay.
and oxygen minus.
Okay. What over here?
2 and minus 2. H2 is 2. And
Plus
You don't multiply. Okay.
plus 1.
Okay. Minus 2. All right. So looking at this, my question again, what is being oxidized? What is being reduced here?
carbon is oxidized.
Okay. Carbon is oxidized. It's increasing its oxidation state. Okay. It's losing electrons. Okay. There are fewer electrons on carbon. Okay. And what is being reduced? Oxygen is being reduced. It goes from 0 to minus 2, so it receives electrons. Okay. So electrons are transferred, formally at least, from carbon to oxygen. Yep.
Which oxygen is being reduced? Like,
Well, at least some of it.
yeah.
Well, obviously this one.
Yeah, but
This one, but
this one gave us which of these two which oxygen? Did it give us the oxygen, the CO2 or the H2?
in this reaction, I don't think you can easily tell. In metabolism, you can tell very easily which one it is, and that's what we're going to be talking about now. When you burn it up, I don't think it's easy to actually figure out. I'm not sure the experiments have been done. I don't know. Okay, because it's at high temperature, everything gets broken down, things react with things, so it could be random. I don't think there's actually any pattern in that. But it's possible that the experiments were done with isotopes and that they actually know, but I don't know. Okay? But for metabolic breakdown of glucose, we do know. Okay? Right. So this is the reaction of burning of glucose. Something similar happens in our bodies, but it's only similar when we summarize it like this. Okay? The actual steps in that and the actual machinery that's involved are completely different. Now, why can't we just perform this reaction in our body just straight away? Yeah.
The extreme heat will burn our cells, tissues, and stuff.
Okay, so one thing, the heat and presumably the light would be quite inconvenient, okay? Because there's too much released of it in one go, okay? Yep. Okay, so if we can split it into separate steps, we can release bits and bits of energy that we can use, correct? Okay. Other reasons? Yep.
The energy wouldn't be able to transport throughout the body, so in
Okay.
case you would, for example, break it down in the liver, you wouldn't be able to use it somewhere else.
Okay, so we wouldn't be able to capture the energy in a useful way, okay, which is too much released, yeah? Sure, okay. We're still talking about the same thing, okay? You're all right, but there's one more reason for this to not happen in our body. We don't, but we still get it anyway, okay? Yep.
Activation?
Okay, you actually have to heat it up for this to happen, okay? That's why when I was talking about burning glucose, you actually have to put a light to it, put a match to it so that it actually sets a light, okay? So at our body's temperature, this reaction does not occur, okay? We can't actually react stuff with oxygen just like that at low temperatures. As we can all see at this point, because we're all sitting in 20% oxygen, okay? And we know that our bodies are actually flammable, okay? If you heat them up sufficiently, they will burn, okay? And probably the wood as well, okay, as we know. But it doesn't, okay? So oxygen, quite interestingly, is quite poorly reactive, okay? For gas, which is extremely electronegative, and I'm not going to go into those details, you've probably heard about electronegativity, oxygen is the second most electronegative element, okay? It wants to work with electrons, okay? It wants to gain electrons, it wants to get reduced, but it doesn't, okay? If we were sitting here, do you know what is the most electronegative element? Fluorine, okay? Now imagine we were sitting here in a lecture hall and there would be 20% fluorine around here, okay? You can probably imagine what would be happening, okay? Our bodies would, either they would burn or they would just gradually get dissolved because the fluorine would react with our bodies, okay? That's why fluorine is a very toxic gas. But oxygen, we're fine, nothing's going on, okay? Because oxygen is quite unreactive. And the reason why it's unreactive, it's not about thermodynamics, it's about kinetics, as you correctly said. And the problem with oxygen is, have you heard what a radical is in chemistry? Can somebody explain what a radical is? Okay, somebody maybe from the back so that I know that you know what it is. Okay, raise your hand if you've heard of it. What a radical, okay, can you explain what it is? Yep.
It's like a highly reactive intermediate. It's like it's got like is it a homolytic electron attached to it? Like
okay.
The electron is broken. Like you have a pair of electrons that's broken. And you have an
okay.
electron.
Okay. So it is a chemical species that has an unpaired electron. Okay. That is the definition of a free radical. Okay. And as you said correctly, most of them are extremely reactive. Okay. Very short-lived species. They want to react with everything. Now, the interesting thing about oxygen is that it's a bi-radical. Molecular oxygen, O2, has 2 unpaired electrons. Quite interestingly, they never pair up, okay? At least at normal conditions. They never pair up. They're 2 unpaired electrons. A very strange species. Now, your colleague said that radicals tend to be extremely reactive. But for oxygen to react with anything, it would have to find a molecule that has another 2 electrons with the opposite spin, so that they can actually pair and form bonds, okay? And those chemicals don't really exist, okay? So for molecular oxygen at normal temperature, it's very difficult to react with anything, as we can see. But as you raise the temperature up, those 2 unpaired electrons in the topmost orbital can actually jump to a higher orbital and they can pair up. And once they pair up, oxygen becomes very reactive. And that's why when we, increase the temperature of something, it will just burn, okay? If it's thermodynamically favorable, okay? So this is just a kinetic aside to our discussion. The other reason, apart from it being inconvenient to release so much energy in one go, the other problem is that this would not occur at room temperature. So we need to split it up, we need to do different kinds of tricks, and we also have to use enzymes to actually allow those things to happen, okay? Right. We can split, now we're going to be talking about metabolic pathways, okay? So let's forget about burning of glucose. Let's talk about the metabolism, the breakdown of glucose in our cells. And we can split the breakdown of glucose and the release of energy that we need to do some work into two phases, okay? In the first phase, what we do is we remove electrons from glucose, okay? So we oxidize glucose. Well, we oxidize glucose by removing electrons, and what we get from it is carbon dioxide. Okay? Where do we put those electrons to? I mean, we need to keep them somewhere. Huh? Okay, we use redox coenzymes, such as NADH. Okay? So I'm just going to put here 2 electrons, because they're usually packed in pairs. Okay? Not for some chemical reason, it's just that's how it works. Okay? NADH carries 2 electrons. Do we know any other redox coenzymes? Okay. Okay? So we have redox couples, NAD plus, NADH, and FAD, FADH2, and they each carry 2 electrons. Okay? This is the oxidized form, and accepts 2 electrons and turns into the reduced form. Okay? This is quite important. Raise your hand if this is clear. Okay, not everybody seems to be on the same page. Okay, so I'll just repeat it. NAD plus is a molecule that can be reduced by 2 electrons to form NADH. Okay, so this is the reduced form, this is the oxidized form. The same thing is for FAD. FAD is the oxidized form. If it receives 2 electrons, it turns into the reduced form. And then obviously it can turn back and release those 2 electrons to something else. Okay, so these 2 are just carriers of electrons around. Okay, because we can't have naked electrons easily in our cells. Yeah.
Is the loss of energy always equal to transferred energy?
Isn't what equal to?
Is the loss of electrons, for example, is FAD like it has more energy? It has NADH2 more energy because it has more electrons than
No.
FAD?
No. No.
H plus H plus.
Let's forget about that. Okay. They both carry 2 electrons.
Oh yeah, no, sorry. I just mean it's the oxidized form because it has more electrons. Is it always the one that has more? That carries more?
No, the reduced form is the one that has more electrons. Okay, because the reduction is gaining of electrons.
And does the gaining of electrons mean gaining energy? Is it like?
No. And actually, I don't want to get too far away from my lecture, but actually it's sort of losing energy, because what we do is the electrons that are in glucose are at some kind of a high level. They're high energy electrons, sort of. Okay, we're going to talk about it, what it means high energy electrons in a minute. Okay, so they're quite high in the potential, and since we need to transfer them to somewhere else, for example to NADH, they have to drop down a little bit. Okay, so that it actually spontaneously occurs, and then they drop down again, and then drop down again. So, in fact, those electrons, when they were still in glucose, they had more energy than they have on NADH. Otherwise, the reaction wouldn't occur. Okay, but I'm not sure it actually answers your question. But okay, you can ask during a break or something. Yep. All right. So in the first phase, we transfer electrons from glucose onto reduced coenzymes, well, onto oxidized coenzymes, making them reduced coenzymes, and release carbon dioxide, okay? And probably some water or something. In this phase, we don't need oxygen, okay? Oxygen is not needed for this part at all, okay? It may seem abstract now, but when you talk about glycolysis and Krebs cycle, it will be much clearer what I mean by this, okay? So for glycolysis and Krebs cycle, you do not need oxygen at all, right? So this is the first phase. Now in the second phase of metabolism, not just glucose, but other things as well, we do need oxygen, and that's the part we're going to be talking about today. And it is that part when we take those reduced coenzymes and we reoxidize them And we reoxidize them by dumping those electrons that we got from glucose onto oxygen. Does it make sense? So in the first phase of metabolism, we just keep removing electrons, putting them onto coenzymes. In the second phase, we take those electrons from those coenzymes and we dump them onto oxygen. And this is the bit we're going to be talking about today. Okay, does this make sense? All right, well, let's now talk about the huge enzyme machinery that's called the respiratory chain or the electron transport chain, which resides in mitochondria and which catalyzes this whole reaction. Okay, right. Okay, what do you think this is? Okay, very good. These are the mitochondrial membranes. Okay, as you know, mitochondria have two membranes, the outer membrane and the inner membrane. And these are the two membranes. Okay, so this is the outer membrane. This is the inner membrane. What is this then? The intermembrane space. Very good. And what is this? It's the matrix. Okay? Now, first important thing, I drew the two membranes as if they were different widths. Okay? They're exactly the same. Okay? I just don't care in what I need to be talking about. I don't care about the outer membrane. Okay? So I just drew it very small. But actually, they are exactly the same width. Okay? So don't think that some kind of a smaller membrane. Okay? It couldn't really be. Okay? All phospholipid membranes have basically the same width. Yeah?
The outer membrane, is it to the cytosol? Like
yeah, here would be the cytosol. Okay, cytosol, intermembrane space, matrix. Okay? Right. The respiratory chain that we're going to be talking about, I'm just going to write it down because I think some of you have not heard it. Okay, respiratory chain or electron transport chain. This electron transport chain resides inside the inner mitochondrial membrane. Okay, it's in there where the whole chain exists. Okay, now it's some kind of a chain. What kind of chain? It's a chain of enzymes, or rather enzyme complexes. Because those enzymes that catalyze the steps in the electron transport are actually huge proteins containing lots of subunits. So we call them enzyme complexes or just complexes. Okay? Now, the first complex in the sequence is called, unsurprisingly, complex 1. Okay? It's called complex 1. And the purpose or the function of complex 1 is to reoxidize NADH. Okay? So it takes NADH and turns it back into NAD+. Okay? Remember we said that in the first phase of metabolism we remove electrons onto redox coenzymes and then we need to reoxidize them. So reoxidation of NADH is what complex 1 does. Okay? Takes NADH and changes it into NAD+. Right. What happens with those electrons? Obviously those electrons need to be taken away and put onto something else. Okay? Yeah? Okay, you with me? Right. So complex 1, again, it's a huge complex, contains 45 subunits, massive protein. It looks completely different than the way I drew it, but it doesn't matter. The electrons go through this massive protein, okay? Lots of different places where it can bind. And then they leave the complex and are bound to something that we call coenzyme Q. Coenzyme Q, CoQ, or ubiquinone. Ubiquinone. Okay? So once again, the reaction that is catalyzed by complex 1 is taking electrons from NADH and dumping them through quite a complicated process, dumping them onto CoQ. Okay? CoQ, most of you have probably heard of it because it's in those commercials for anti-wrinkle creams. Okay? CoQ10. This is what we're talking about. Okay? So this is the coenzyme that is the acceptor of electrons from NADH in the reaction catalyzed by complex 1. All right? Okay. Now, in this process, when ubiquinone receives the 2 electrons, we're still talking about 2 electrons in one packet, okay? So 2 electrons leave NADH, they're dumped onto CoQ. In this process, ubiquinone is reduced to ubiquinol, okay? Okay, so basically a ketone, well, it's a quinone, but anyway, a ketone is reduced to alcohol, ubiquinol. Okay? So this is what happens in complex 1. Now, complex 1, usually when you read literature, you'll find it's a respiratory complex 1. That's what everybody calls it. But there are other names for it that are more systematic. One is NADH oxidase. Okay? Because it oxidizes NADH. But the most systematic name for it is NADH ubiquinone oxidoreductase. Okay? Oxidoreductase is an enzyme that takes electrons in one place, puts them in another place. So oxidoreductase, a general name. And now we need to tell the reader where are the electrons being taken from and where are they being put onto. Okay? So it's NADH ubiquinone oxidoreductase. Yep? No, it's NADH dehydrogenase. Okay? Because that's what's being dehydrogenated. Okay? One thing about the way I kind of conceptualize this whole area, if you could just, yeah. Dehydrogenase is a word that I'm not going to use because I think it's quite confusing because it makes you think that what is being transferred are hydrogens. Okay? I don't want you to think that. I want you to think about electrons. Okay? That there are some hydrogens, like you said, there's some H plus and whatever. Forget about it. It's not interesting for the chemistry at all. Okay? What is interesting is that electrons are being transported. Okay? Some H plus ions will always bind to it. Not interesting at all. Okay? Electrons are what matters. Okay? So NADH dehydrogenase, yeah, it's something you'll find in the literature. I just don't want you to use it. Okay? Okay? So it's NADH ubiquinone oxidoreductase. Right. Okay, so we've reduced our, by the way, CoQ is what we call a small molecule. It's not a protein, okay? It's not a protein. It's called a small molecule. It's actually a pretty huge molecule, but it's not a protein. And it's extremely insoluble in water, okay? It's extremely hydrophobic. So it actually resides inside the membrane because that's the only place that can dissolve it, okay? It can't really exist in the water compartment that's outside the membrane because it's extremely insoluble. Okay, so it basically sits inside the membrane and just moves around the membrane. It's dissolved in the membrane in those phospholipids. Yeah.
Is ubiquinone turned straight away to ubiquinol or has a
in
between lines?
Yes, it does. It's called semiquinone. I'm not sure where you're heading with that answer.
If it has something to do with the complex 1.
Yes, but I don't want to complicate it too much. Okay, so we transfer 2 electrons from NADH to CoQ. In fact, you can transfer just 1 electron to CoQ to make a semiquinone, and it will become slightly more important in complex 3. But yeah, I think it's maybe too much information, but it is possible. Yeah. Okay, so CoQ, a mobile carrier of electrons inside the membrane. Right. Since it is an electron transport chain, we need to move those electrons to the next step in the chain. And the next step in the chain, and now it's slightly less logical, is complex 3. Complex 3. Okay, so ubiquinol, the reduced form of CoQ, moves to complex 3 and dumps the 2 electrons onto complex 3. In this case, it actually dumps them one at a time, so it goes through the intermediate, which is called semiquinone. But it's not really that important for you at this point, okay? So let's just take that both electrons from ubiquinol are dumped onto complex 3. Something happens to those electrons inside complex 3. And then they are dumped again onto something else, which is another mobile carrier called cytochrome C. Cytochrome C. Yep.
Isn't cytochrome C supposed to be in the blood since it has heme?
No. Not everything that has heme has to be in the blood. Okay? Hemoglobin has heme, correct? But there are lots of other things, for example, cytochromes, that are not in the blood and they do have heme as well. Okay? Heme is a generic coenzyme or prosthetic group or whatever you want to call it that is in many different kinds of proteins. Okay? So hemoglobin is one hemoprotein, but cytochrome C and other cytochromes are other hemoproteins that are in mitochondria or in the endoplasmic reticulum, what have you. Okay? Right. So this is the reaction that is catalyzed by complex 3, another big enzyme complex, takes 2 electrons from CoQ and transfers them to cytochrome C. But unlike in complex 1, or rather CoQ, cytochrome C only receives 1 electron at a time. Okay? So the 2 electrons that come from CoQ have to be split in a way in complex 3, and then one at a time they are added to a molecule of cytochrome C. So cytochrome C only receives 1 electron. So we started with 2 electrons on NADH, there were 2 electrons in complex 1, 2 electrons on Q, and then they are split in complex 3 to 2 separate electrons, and one is dumped to cytochrome C and then another one is dumped to another cytochrome C. Okay? Cytochrome C, as was already mentioned, is a small protein that contains heme. Heme as a cofactor, as a prosthetic group. Does everyone know what heme is? Raise your hand. Heme, yeah? Okay, about half the people. So heme is quite a complex organic molecule. You're going to talk about the metabolism of heme, the synthesis of heme quite soon. And it contains iron, an atom of iron inside, okay, which can receive electrons and dump electrons as well. So it's redox active. Okay? You've probably heard in chemistry that we have 2 forms of iron, well, 2 forms of ionic iron, and it's Fe2 plus and Fe3 plus. Okay? And the iron can cycle between these 2 forms. Okay? How does it cycle? Well, if it receives an electron, it's reduced from 3 plus to 2 plus, and if it gives away an electron, it moves from, it's oxidized from 2 plus to 3 plus. Is this familiar? Yeah? Very good. So this is what's happening in cytochrome C, only the iron there is not just floating around, but it's enclosed in heme. Okay? So it's a hemoprotein that can cycle between these 2. And since you can only use 1 electron to reduce iron 3 to iron 2, it only carries 1 electron. Yeah? Yeah? Sort of, maybe?
So
not really.
Each cytochrome C contains 1 heme group?
Correct. Each cytochrome C only contains 1 heme and therefore can only receive 1 electron.
And is it actually bound to the inner membrane, the cytochrome C?
It's in the intermembrane space, but it's attached to the inner membrane.
Not bound?
It's only electrostatically, it's kind of, it sticks to it, okay? But it's not bound in covalent way or anything, okay? So it's in the intermembrane space, but actually sits on the membrane, okay? Yep.
In the time between the two electrons, like, does another cytochrome C come? Or?
Sure, yeah. There are plenty of molecules. In the inner mitochondrial membrane, there are loads of molecules of complex 1, loads of Q molecules, many molecules of complex 3, many molecules of cytochrome C, and they just kind of move around on the membrane. Okay? So yeah, absolutely. So one cytochrome C takes an electron, goes away, another comes, takes an electron, goes away, another Q comes, and okay? You can imagine that the things are just moving in the membrane, okay? This is a cross-section, but if we actually look at it from the top or from the bottom, we would see lots of different complexes and molecules moving around between them and transferring electrons, okay? So this looks like a static cross-section, but actually we're just looking at a very small part of the membrane, okay? There are lots of molecules of all of these components in the actual membrane, okay? And they can move around because it's a fluid membrane, okay? Yeah.
I thought this one was in hemoglobin, and in the cytochrome goes from 2 plus to 4 plus.
No. In cytochrome C, it goes from 2 plus to 3 plus to 3 plus to 2 plus. Okay? In cytochrome oxidase, which is going to be the next step, but I'm not going to be talking about iron 4. It's just, yeah, we can talk about it later on if you're really interested. Okay? But I just want to keep it quite simple, the chemistry. All right? Okay, what is going to be the systematic name for complex 3? In the same way as we made one for complex 1.
CoQ cytochrome C oxidoreductase or something?
Okay, so you could say CoQ cytochrome C oxidoreductase, but we really want to also tell the reader or the audience what are the redox states of those two substrates. So CoQ has 2 redox states, okay? It's got a ubiquinone and ubiquinol, okay? One is oxidized and the one is reduced. So which one is it going to be in this case? It's going to be ubiquinol, okay? Because we need to take those electrons from the reduced form, because the oxidized form does not have any, well, it has electrons, but I can't give them away. Okay? Okay, raise your hand if you lost me, or I lost you rather. Okay, good. Okay, so what I'm just saying, you could name the enzyme by just saying that, well, there's some CoQ and there's some cytochrome C and electrons are transferred between them. And in a way, that's fine. But I really want you to specify which redox form, whether the oxidized or the reduced, is the source of electrons, and which form of cytochrome C, whether the oxidized or reduced, is the acceptor. I mean, it's kind of logical because we're moving electrons, so obviously it's going to be from the reduced to the oxidized form. But I want you to put it in the name. So you correctly said that this is going to be ubiquinol, that's the reduced form. Okay? How do we do it with cytochrome C? Well, we have 2 forms of cytochrome C as well. One has 2 plus and the other one has 3 plus. And we call them ferro cytochrome C and ferri cytochrome C. Okay, which one's which? Okay, so ferro is the 2 plus form, the reduced form, and ferri is the oxidized form, the 3 plus form. So, how's it going to be, the whole thing, for complex 3? Ubiquinol, ferri cytochrome C, oxidoreductase. Okay? We take those electrons from ubiquinol, we put them onto ferri cytochrome C, thereby changing it into ferro cytochrome C, and it's called oxidoreductase.
Do you repeat it all nice?
Yep. Ubiquinol, ferri cytochrome C, oxidoreductase. Oh, well, I can, maybe. So this is 3? Yes. Well, they're pretty simple. Once you know what happens, it's quite easy to make them. But anyway. Okay, so this is complex 1. NADH, ubiquinone, oxidoreductase. Yep. Right. Okay, so we've managed to put our electrons onto cytochrome C. And we need to move them further because remember the whole idea of the respiratory. Do you still have more questions about that? Do you have any question? No? Yeah? You do seem to have a question. You sure? Okay. Remember that the whole idea of the respiratory chain is to take electrons from NADH and some other coenzymes and put them onto oxygen. So in the last step of the respiratory chain, we take those electrons from cytochrome C and we put them onto oxygen. And in order to do that, we need another enzyme complex, which in this case, we're kind of returning to the logic, is going to be complex 4. Okay, complex 4. Which takes those electrons one at a time from cytochrome C, from reduced cytochrome C, so ferro cytochrome C, and through quite some complex chemistry, dumps them onto oxygen, thereby forming water. Okay, reducing oxygen into water. Okay, once again, I don't care about those hydrogens there, okay? There are plenty of hydrogens around, not interested, okay? What I need is to reduce oxygen from 0 to -2, and then those hydrogens just come and form water, okay? Not interesting. Okay, how many electrons do we need to reduce one molecule of O2, one molecule of oxygen, into water?
4.
Okay, who said 4? Okay, correct. Okay, we need 4 electrons. 4 electrons need to be dumped onto O2 in order to form 2 molecules of water. Okay, remember in water, oxygen has -2 oxidation state. So you have 2 molecules of water, we need 4 electrons in total. So even though cytochrome C only carries 1 electron, complex 4 will basically wait for 4 of them to come. It will accumulate 4. And then it will form water. Okay? A very interesting process that involves some exotic oxidation states of iron, including probably iron 5 as well, but let's not complicate it. Okay? So 4 electrons sequentially have to come from cytochrome C in order to make water from 1 molecule of oxygen. And that's it. That's the electron transport chain. Yeah?
Are the electrons left in the complex 4, or do they bind one by one to oxygen?
Quite a complex process. There's a very interesting called binuclear site. There is a copper atom and there is a heme, and the oxygen is kind of spread between them, the two atoms of oxygen, and sequentially 4 electrons bind to it until it splits and forms water. Very interesting, but I think far above what you need to know. Okay. So this is basically the electron transport chain. Yes? Yep.
I have a question. You said that at the cytochrome C,
Yep.
it receives 1 electron,
Yep.
and another cytochrome comes to receive
Correct.
the electron.
Correct.
Yeah, but when it receives the electron, it keeps it immediately to complex 4.
Well, immediately.
It accumulates 4 cytochrome C over the between complex 3 and complex 4 to receive.
Once again, there are lots of molecules of complex 3, lots of molecules of complex 4, lots of molecules of cytochrome C. So they just move around, and if they happen to come to a reduced complex 3, they will take an electron. If they happen to come to a oxidized complex 4, they will dump the electron. Okay? It's a statistical process. It's a random process, basically. Okay? Yeah?
The systematic name of the 4 complex?
What is?
The systematic
Yeah. What is the systematic name? Yeah? Since you asked. Yeah.
Ferro C.
Ferrocytochrome C. Cytochrome C. Yeah.
Cytochrome C.
Yeah. Oxygen.
Oxygen oxyreductase.
Oxidoreductase. Okay? So ferrocytochrome C, oxygen, or dioxygen if you want to call it specifically, ferrocytochrome C, dioxygen, oxidoreductase. Super simple. Ferro, because now it's reduced. We need to take the electron from the reduced form. Okay? So we're giving the electron to the oxidized form, but now we're taking the electron from the reduced form. So it's ferrocytochrome C, dioxygen, or oxygen, oxidoreductase. Yep?
So from the first complex we got 2 electrons, then we pass those electrons from the equinol to cytochrome C. So we have two cytochrome Cs for each equinol.
Cytochrome C. Yep.
Cytochrome C for each equinol. And then we have to wait for four cytochrome C to make two others.
Exactly. So we started with one molecule of NADH, which only carries 2 electrons. So in order to complete this whole thing, we actually need 2 molecules of NADH to go through this, okay? Which in the end will mean 4 molecules of cytochrome C that will go to complex 4 and will form water. Yep.
And FADH enters somewhere else.
Yeah. I'm just getting to it, okay? So do you all understand this simple thing? This is how we, well, simple, it's not simple, but do you all understand that this is how we reoxidize NADH in our mitochondria, okay? Complex 4 is ferrocytochrome C, oxygen, or dioxygen, oxidoreductase, okay? Right. Now, the question, what happens to FADH? Or FADH2 rather. First of all, FADH2 never occurs in solution just as a naked coenzyme. Unlike NADH, NADH is a simple molecule that just moves around, it's dissolved, okay? It's a simple naked coenzyme. FADH2 always exists inside a protein. It's never dissolved outside, okay? So rather than talking about this hypothetical thing, FADH2, let's just talk about the enzymes that contain it, okay? Again, in literature and elsewhere, you will find people talking about NADH and FADH2 or NAD plus and FAD. But those are completely different things. While NADH exists on its own, FAD is always bound to a protein. Okay, so the question was, what happens to FADH2? Well, I'm going to answer in a slightly different way, in a slightly different concept. Here, the entry point for electrons is complex 1, which takes those electrons and dumps them onto Q. But in addition to complex 1, there are other sources of electrons that can also, other enzymes, that can also dump their electrons onto Q. Okay? And some of those enzymes do contain FAD in them. Okay? So this is sort of the answer to what happens to FADH2. Well, it's reoxidized by taking those electrons and putting them onto Q. But this FADH2 is always bound to an enzyme. And one of those enzymes that does exactly that, so it takes electrons from something else and dumps them onto Q directly without going through complex 1, is an enzyme of the Krebs cycle, of the tricarboxylic acid cycle, citric acid cycle, whatever you want to call it, which is called succinate dehydrogenase. Okay? So imagine, in addition to all of this, we have another enzyme here, which is the enzyme of the citric acid cycle called succinate. What does it catalyze? Does anyone know? Okay, so it oxidizes succinate to fumarate. Okay, it's an enzyme of the Krebs cycle. Incidentally, it's also present in the inner mitochondrial membrane. It sits in the membrane. Okay, it catalyzes the oxidation of succinate to fumarate, and since it is an oxidation, it itself, the enzyme, has to be reduced. Okay, remember always when there's oxidation, there's always reduction. Okay, so succinate is oxidized to fumarate, and the enzyme itself becomes reduced. And in order for the enzyme to work again, it has to be reoxidized as well. How does it become reoxidized? Well, it just gives those electrons to Q. And Q then carries them to complex 3, etc., etc. Okay, raise your hand if this makes sense. Because I can see that most of you are just blank.
How many electrons?
The 2 electrons. Okay? So 2 electrons are taken away from succinate to form fumarate, and those 2 electrons through succinate dehydrogenase, which incidentally contains FAD inside, okay, are dumped onto Q. Yep.
So that's there are more ways to give electrons to the electron transport chain.
Correct. Okay. Complex 1 is one of them, but for example, succinate dehydrogenase is another way to enter the electron transport chain. Okay? And there are other enzymes and other proteins that can also give electrons directly to Q. One of them is called, for example, I can't draw it here easily, so I'm just going to put it here. It's called ETF, which stands for electron transferring flavoprotein. It's a small protein that takes electrons from something called beta oxidation, and you're going to hear about beta oxidation very, very soon. It's the metabolic pathway for degrading fatty acids. Okay? And electrons, at least part of the electrons from beta oxidation, are carried by this protein called ETF, and also those electrons are dumped directly onto Q. ETF, since it is a flavoprotein, also contains FAD. Now, final bit before I answer the question. Succinate dehydrogenase, the enzyme of the Krebs cycle that we just talked about, is for historical reasons called complex 2. Okay? So this alternative way of, one of those alternative ways of putting electrons into the electron transport chain, which is succinate dehydrogenase, is also called complex 2, which kind of completes the logic of the whole electron, at least of the naming. Okay, so we have complex 1, complex 2, complex 3, complex 4. But this complex 2 does not really take any part in the pathway from complex 1 to complex 3 to complex 4. It's just an additional thing that also can give electrons to Q.
And why does it yield less ATP then?
We'll get to that in the second half of the lecture. Okay, so let's have a 3-minute break.
Is the FAD thing related to the NAD?
Okay, the first question is the FAD thing related to NAD thing? Okay, not really, okay? We need to reoxidize, let's say, 2 forms of electrons. One is in NADH, and the other one is in proteins that have FAD in them, okay? For NADH, we use complex 1 that transfers those electrons onto Q, and then complex 3, complex 4, and onto oxygen. For FAD-containing proteins, such as succinate dehydrogenase or ETF or what have you, those also, similar to complex 1, they dump electrons onto Q that goes to complex 3 and complex 4. Okay? So there are 2 separate pathways that converge on Q, basically. Okay, other questions? There were quite a few more. Okay, can I repeat it? Right. We have 2 different inputs of electrons into the electron transport chain, and they all converge, or they both converge on Q. Okay, Q is their common factor, and from Q, there's only one way. Okay? The first one is complex 1, which takes its electrons from NADH and then moves them onto Q. The other input, or the other group of inputs, is from, for example, succinate dehydrogenase, ETF, and other enzymes that contain flavin in them. They contain FAD in them, but it's not really interesting that they do, but they do, okay? And they also dump their electrons onto Q, and then it follows the same way. Now. Okay, let me draw a better picture. Okay. Okay, so this is the sequence of the respiratory chain starting from Q. Q, complex 3, complex 4, oxygen. Now, to Q, we can start with complex 1, into which electrons from NADH go. Or, we can put electrons onto Q from succinate dehydrogenase. Or from ETF. Or some other enzymes. Still not better. Is it? Okay. Okay, there were some other quite important questions. Oh, what happens, why do you always see that it's NADH plus H plus that turns into NAD plus, okay? For me, that's not important, okay? It's always there because you want to keep the reaction electron neutral. You don't want to be losing charges, okay? So if you start with a positively charged thing and you don't add any negatively charged thing, you have to end up with a positively charged thing as well, okay? So you balance the equation by adding an H plus. Does it make sense? Okay, this is the simplest explanation. It could be explained in a more complicated way because when we draw the NADH here, we can say that what is transferred is actually a hydride anion. It's H minus, so 2 electrons plus H plus. But to me, it doesn't matter if you write those H plus there. What I'm interested in, as I said, are electrons. I don't care about protons. I don't care about hydrogens. Okay, they're not interesting at all. Yeah.
As there's many ways to get to electrons to Q. Do we have another type of electron giving enzymes to cytochrome C? Like, do we have something else that gives electrons to cytochrome C apart from Q?
As far as I know, we don't. Mammals definitely don't. There may be something in plants or something, but I doubt it actually. I don't think so. I don't think there are any. Definitely not in our body. In other animals or stuff, possibly. But as far as I know, there aren't any. No. But you can do it artificially. There are artificial ways of putting electrons onto cytochrome C if you're so inclined. But yeah. Okay. Other questions?
Could you please have a quick repetition of what happens when the succinate turns into fumarate and like?
Oh, I thought I did, but that's fine.
No,
Okay.
like in the complex too.
Yeah. So succinate in complex 2, succinate dehydrogenase, succinate is oxidized to fumarate, and 2 electrons are taken from it and put onto Q. That's what succinate dehydrogenase does. Yep. Other questions? I think there's some other ones as well that were quite important, but nobody wants to ask anymore. Okay. Yeah.
So FAD is bound to succinate, or is it part of succinate?
No. FAD is part of succinate dehydrogenase, part of the enzyme. It's inside the enzyme.
So the enzyme gets oxidized as well?
No, the enzyme gets reduced. Because we oxidize succinate, the enzyme gets reduced. And it needs to be reoxidized by Q. Okay? Right. What is the reason, continuing with the lecture, what is the reason that electrons flow from NADH all the way to oxygen? What makes them go that way? Yeah?
Oxygen is electron negative, like boost on that?
Okay, so one explanation is that oxygen is very electronegative. It likes electrons, okay? But electronegativity, do you have a question or you want to?
I was going to answer, but I also had a question.
Okay, so let's finish with this, okay? So electronegativity is something that explains the affinity to electrons, but I want to use a slightly different concept, which is called redox potential or reduction potential, okay? I'm going to call it reduction potential. I think it's less confusing. Reduction potential. So it's how easily something gets reduced, how easily it accepts electrons. Raise your hand if you understand what reduction potential is. Okay? It's a number that tells us how easy it is to reduce something, to give electrons to it. Okay? Reduction potential or redox potential. Right. So what happens here is we're actually moving from a relatively low redox potential or reduction potential to a very high reduction potential. Oxygen, as you correctly said, with electronegativity, but that's a slightly different concept. Oxygen really likes electrons, okay? It has trouble with reacting with them, as we said in the beginning, but that's a kinetic problem. Thermodynamically, it really likes electrons. NADH, on the other hand, doesn't really like those electrons very much. It actually likes them more than glucose, because otherwise the reaction wouldn't occur, okay? So NADH has enough affinity for electrons to pull them away from glucose, but it has much smaller affinity to electrons than oxygen, for example, has. Much, much, much smaller. So what pulls or what pushes those electrons through the electron transport chain is increasing reduction potential. So we start with NADH that has minus 0.3 volts or something like that. You don't have to remember that, okay? Then the first step in the enzyme has a higher affinity, okay? So higher reduction potential. And then it has even higher, Q has even higher, complex 3 or bits in complex 3 have even higher reduction potential, cytochrome C has even higher reduction potential, complex 4 has even higher, and oxygen has the highest reduction potential. That's where those electrons end up, okay? So this is what pulls those electrons through the electron transport chain. Now, it's slightly confusing, but if you can imagine a cascade of dams, the water flowing from the highest dam to the lowest dam is basically moving along a gravitational potential, moves down in the gravitational field, okay? Here you can imagine those electrons flowing exactly the same way along a reduction field or something like that, or redox potential field, okay? The trouble here is that the reduction potential is going up, so actually they're flowing up, but do you get the analogy with water? Okay, so the electrons want to go to as high reduction potential as reasonably possible. So this pulls them or pushes them through the electron transport chain. Now, the same way as in dams, we can use this affinity of water to low gravitational potential, if you want to, if you know what I mean. Okay, so we can use the water trying to get down to do some work. For example, make electricity. We can do the same thing here, and actually our cells do that, our mitochondria do that. What kind of work do we turn these differences in redox potentials into? Does anyone know? ATP will come in the end, but correct. We use this pull of electrons towards oxygen to pump protons from the inside of the mitochondrion, from the matrix, into the intermembrane space. Okay, so complex 1 pumps protons, sorry. Complex 3 also pumps protons, and complex 4 also pumps protons into the intermembrane space. Nothing else pumps protons. Okay, only complex 1, complex 3, and complex 4. And this is the answer to your question, why FAD has less energy or something.
Because there's less hydrogen pump?
Well, because you miss one hydrogen pump, okay? But, okay, I don't want to confuse other ones who did not have that question, okay? And it actually has to do with redox potential as well, because FAD has a higher redox potential. Let's not confuse it too much. Right, the important thing is, the difference in potentials between NADH and Q is used to pump protons in complex 1. The difference in potentials between Q and cytochrome C is used to pump protons in complex 3. And the difference in potentials between cytochrome C and oxygen is used to pump protons in complex 4. Okay? Right. So the energy, the potential energy in those differences in redox potentials is transferred, is transformed into another form of potential energy. And in this case, it's the concentration gradient of H+, of protons. There are many, many more protons here in the intermembrane space than there are in the matrix. Does it make sense? Really? Raise your hand if that makes sense. Okay, I think you just raise your hands every time I say raise your hand. Okay. All right. Okay, so we take one form of potential energy, which is the difference in redox potentials, and we turn it into another kind of redox of potential energy, which is different in concentration, difference in concentrations. And since we are talking about a difference in concentrations of a charged species, we're talking about protons, H+ ions. The difference in the concentration also can be represented as difference in electric potential. Okay? There's going to be a potential difference, an electric potential difference. There are more positive charges here than there are here. So there's going to be voltage between the two bits. Make sense? Yeah? So we can use this voltage, or this gradient, or concentration difference, to make other things. To make the same way as we can use a battery to light something, or to have an electric engine, to move an electric engine. We can use this potential difference to make some other kind of work and do something interesting. And what we do is we synthesize ATP. So we use this potential difference to synthesize ATP. Okay, how does it work? Well, there is another enzyme in the inner mitochondrial membrane, which is called ATP synthase. So it's an enzyme that synthesizes ATP. It's a pretty simple name. It's called ATP synthase. And this enzyme allows those protons, and there are many, many more of them here. As we said, there are many more of them in the intermembrane space than there are in the matrix. It allows those protons to flow back into the matrix and uses the of those protons that really want to go back to make ACP. Okay? How does it do it? It's a very interesting enzyme. It actually, as those protons flow through the enzyme, it physically starts turning around. It really looks like a turbine. Okay? The actual protein molecule turns around, well, this part anyway, turns around, and in the movement, it basically pushes ADP and phosphate together and forms ATP as it moves around. Okay? Very interesting. If you have time, just put into YouTube, put ATP synthase or something like that. There are lots of videos that actually look exactly what the enzyme does and how it does it. It's quite amazing. John Walker, who discovered the mechanism, got a Nobel Prize for it. It's pretty cool, I think. Okay? So this is ATP synthase, which uses the gradient, the proton gradient, to make ATP. Yeah, just 1 second. From ADP and phosphate to ATP. Yep.
Is it correct to say that ATPase turns the energy in the formula potential to mechanical energy?
It does. And then the mechanical energy, or the movement, is turned into chemical energy again in the form of ATP. Okay? So that would be correct. Yeah. Now, if it looks like, you know, how does just the difference in concentration, how can it turn something? How can it, you know, make something interesting? Well, if we transform the concentration difference into voltage, as I said, is possible. The voltage on the inner mitochondrial membrane is approximately minus 150 millivolts. 150, 180, depends on how well the mitochondria are working. So we're minus 150 millivolts. Is that a lot or not so much? Why do you think it's a lot?
It's higher than the usual membrane.
Okay, so it's definitely higher than what our cell membranes, for example, neurons have, correct? But it's also pretty high if you imagine how thick the membrane is. Does anyone know how thick the membrane is, approximately? Yeah? Yeah, about 6 or 7 nanometers or something like that. So you have a potential difference of minus 150 millivolts separated by only 6 nanometers of space, which is equivalent approximately to about 30 million volts separated by 1 meter. Okay? So the force that is trying to push those protons back is immense. It's huge. Okay? And that's why the protein can use it to actually do pretty difficult stuff, which is putting ADP with phosphate together and making ACP. Okay? It's a massive force. Okay? It's actually quite amazing that the membrane withstands this massive electric field. I think it's pretty amazing. Okay? All those proteins have to sit in this huge electric field. I think it's pretty cool. Anyway. Okay, so this is how we use the potential energy in the redox potential difference. In the end, we turn it through a concentration gradient into ATP, into chemical energy of ATP through ATP synthase. Okay? Yes?
At the outer membrane, there's a huge concentration difference between H plus outside and inside.
It is across the inner mitochondrial membrane.
Yeah, sorry.
Okay?
Yeah.
Okay? This is the outer membrane. We don't care about that one. The difference is between intermembrane space and matrix. Yep.
And my complex 1, 3, and 4 help me to get our H plus into the intermembrane space.
Correct.
But from where exactly comes the energy? I didn't get that.
The electrons don't really want to be on NADH. They really, really want to be on oxygen. They're trying to get to oxygen. And we use this force that pulls them onto oxygen to pump those protons there. Or are you asking about how it works in the enzyme?
Yeah.
Okay, well, for complex 1, we don't really know, okay? Well, we're starting to understand that. In the other ones, it's far too complex to talk about it now, okay?
But they work just through.
It basically, you can imagine that, sort of, yeah. Basically, you can imagine that the proton binds here, okay? And as the electron flows through the protein, there are various conformational changes which allow the proton to bind through and then it's spit out in the end, okay?
That was.
Very, very complicated, but yeah, sort of, that's the idea, yeah?
Is the potential over the inner membrane always 150?
Yeah, I mean, if we have mitochondria that work well, it's approximately minus 150, minus 180. It's not that easy to measure, but around that.
It's maintained by when H plus is passing through ATPase, all the H plus is pumped in.
That's right. So if we have enough oxygen and if we have enough substrate, the electron transport chain will just keep pumping and will. Actually, there are regulatory pathways that try to keep the potential at this level. Okay, so if we do something to it, the cell will try to re-establish the potential. So yeah, so it's trying to be at approximately this value. Yeah.
And like in the complexes, when the electrons jump from one thing to other, doesn't it lose energy?
It loses its potential energy. Yeah, absolutely.
Okay,
So it starts at a very high potential, actually low potential. Okay, it works the other way for redox potential. So it starts at some potential and it slowly drops down. So it loses its potential energy. But energy cannot be lost. So the energy is transferred to other things. For example, the conformation of the protein which allows you to pump protons through. Okay, so energy is not lost. It's the electron is losing potential energy, and the energy is transformed into some other kinds of energy.
So can you say that it's recharged when it goes from one complex at the beginning of one complex, at the beginning of the?
No, it's actually through as it goes through the protein. Okay, again, it's massively 45 subunits. It's very, very complicated. Okay, right. I need to speed up a little bit. We only have 5 minutes left. Okay. So this is how we use the potential difference, the concentration difference, to make ATP. But in some of our cells, there are proteins that can also allow protons, and I hope I'm not confusing it with proteins and protons, okay? So there are some proteins that can allow protons to go back into the matrix, and they do not synthesize anything. They basically just make a hole into the membrane and allow protons to go through. Okay, what happens in that case? What happens with the potential energy stored in this potential difference, yeah? Okay, it is lowered, but energy cannot be lost, so it has to be transformed into something. Okay, it turns into heat. Okay, ATP synthase takes this energy and turns it into work. Okay, this is work. Making ATP is work. Okay? But if we just make a hole in the membrane and allow those protons to go back, the energy, the potential energy, is going to be turned into heat. Okay? And as your colleague already mentioned, those kind of proteins that do that are called uncoupling proteins. Uncoupling proteins, they're abbreviated UCP. Uncoupling proteins. Why uncoupling? Well, don't think I have time to explain it, but basically they uncouple the electron transport chain from the synthesis of ATP. So normally in a well-functioning mitochondrion, the transport of protons is always coupled to the synthesis of ATP, as we saw. But if we make a hole through the membrane, we actually dissipate the potential difference in a different way, and therefore ATP cannot be synthesized. That's why they're called uncoupling, because they uncouple the two processes. So for historical reasons, where they didn't understand how mitochondria work, they only could observe that ATP was synthesized, and that oxygen was consumed. Actually, that was the only thing they could measure. So they would always see if oxygen is consumed, we always produce ATP. Okay? But if you make a hole through the membrane, this is not going to work. You're still going to be consuming oxygen, but you're not going to be making any ATP. And this is why they're called uncouplers, because they uncouple the two processes. It's just a terminology. Anyway, so uncoupling proteins allow protons to go back into the matrix while producing heat. The first uncoupling protein discovered was UCP1, and UCP1 is present in brown fat tissue. Okay? And you've probably heard about brown fat, what it actually does. It is now, it seems that it's present in humans, even in adult humans as well, but its precise function is unclear. But many, many animals have lots of brown fat, and they use it for hibernation. So in the winter, when they're sleeping, they can't really move around and produce heat the way we mostly produce heat, by moving around or shivering or something like that. So they use the fat stored in brown fat to produce heat by this mechanism, and it's via UCP1. Yeah?
So different tissues or cells in the body will produce either more ATP or more.
Yes, but the only tissue that can actually do it like that is the brown adipose tissue. So UCP1 is only present, is only expressed in brown adipose tissue. There are other UCP proteins, I think we're now up to UCP5, but their precise function is unclear. Nobody really knows. They're not there to produce heat, okay? They're expressed in many, many tissues, and nobody really knows what they do. There are some hypotheses, but it's not clear what they do. So UCP1, only in brown adipose tissue, and the purpose of it is to produce heat. The other UCP proteins, we'll see what they actually do.
What was the name of the first discovered uncoupled protein?
UCP1.
Oh.
Okay, historically they called it thermogenin as well. I don't think you need to know that name, okay? UCP1 is good enough.
So mitochondria in the brown adipose tissue has more uncoupled UCP.
Absolutely. Well, I don't know if they have more of that or not, okay? It depends on how many protons can flow through. Yeah, I don't know about the amount, okay? And also, even in the brown adipose tissue, it's not there all the time. It's inducible. So if the animal needs to produce heat, it will turn up those AUCP proteins so that there are more of them and they produce heat. If it doesn't need to produce heat during summer, they're turned down and it doesn't produce any heat, okay? So they're not there all the time, yeah?
If you're ruining the proton gradient, why is it producing heat?
Well, because in the gradient, there's some potential energy stored, okay? And we can't get rid of the energy. It has to stay somewhere, okay? And either it can be turned into work or it's just released as heat, okay?
I don't know how it releases heat.
It just heats up the thing.
Okay.
You can sort of imagine, okay, imagine that those protons in the intermembrane space are like ball bearings very high above the ground. And either they can drop through some mechanism that lifts some weights or something like that, or you just drop them like this, okay? And obviously they're going to fall very, very quickly down to the floor because the difference is huge. So you can imagine that those protons, when they're not hindered by these pesky ATP synthesis or something, they just hit the matrix very fast and heat it up, okay? It's not how it works, but I think it's a good analogy. Okay? Right. So UCP proteins, let's just take 5 more minutes if that's okay. UCP proteins, there are also synthetic uncouplers. So there are synthetic molecules, very simple molecules, that can do exactly the same thing. So they don't really make a hole into the membrane, but they can transport protons from one side to the other. So they're called synthetic uncouplers. And the simplest one and the oldest one is dinitrophenol. Super simple molecule. Dinitrophenol, you have phenol with 2 nitrous groups. Super simple. But what it does is it dissipates the potential in our mitochondria. It uncouples mitochondria. Now, why do I talk about it? It was actually used, it was discovered by accident in 1920s because it was in some paints or something and then they found that. Anyway, it was discovered in the 1920s and it was used to treat a disease. What kind of disease do you think it might be used to treat? So it works the same way as uncoupling proteins, okay? It uncouples the potential and turns it into heat. Yeah?
Some people have extra charges in their brain, maybe, and not kidding, extra electric charges in their brain. And
the trouble with electric charges in the brain is that they have nothing to do with protons, okay? They are actually caused by sodium and potassium, okay? These uncouplers can only dissipate a proton gradient. So we're talking about mitochondrial membrane. What do you think, what disease can this treat?
Does it have anything to do with hypothyroidism?
Nope. Okay, it was used to treat obesity. Right? Because whatever energy, potential energy we get from glucose and fatty acids goes through this whole thing and is turned into ATP. That's the purpose of our metabolism, or at least the catabolic part of the metabolism, okay? We use the energy stored in nutrients to make ATP. Now, if we uncouple this process, and the energy is turned into heat instead of ATP, we just keep burning more and more nutrients because the body can't use the energy.
Don't you get a fever or something?
Well, that was the trouble with it, okay? It's extremely effective. People lose a lot of weight, but a lot of them die, okay? Those are very, very, very toxic chemicals, and getting the dose right so that you still keep losing weight, but you don't actually die, is a bit of a problem. One reason was that they were overheating, but the other reason was that if you uncouple the ATP synthesis, some of your organs, such as heart, don't have enough ATP, and they can't actually keep functioning. Okay, so getting the dose exactly right actually proved to be extremely difficult, and at least dinitrophenol was banned very, very soon after they discovered that it's pretty dangerous. So interestingly, you can still buy it on the internet as weight loss pills, but I would stay away from it because it's pretty dangerous. Nowadays, there are much better, much more effective uncouplers that are also available commercially, but none of them are used in clinical medicine because people still haven't found an uncoupler that would work exactly the right way. So they will allow you to lose weight, but they would keep you alive, okay? So uncouplers, quite interesting historically, but nobody, and there are actually lots of pharmaceutical companies and research groups that are trying to do exactly that. So they're synthesizing more and more uncouplers and trying to figure out how to do it. But so far, nobody's actually come up with anything worth using, yeah?
You said uncoupling proteins were also synthetic proteins?
No. Uncoupling proteins are natural proteins that are coded in our genes, okay? Those are synthetic uncouplers.
Those are synthetic uncouplers?
So they're not proteins. They're small molecules, okay? Dinitrophenol. Super simple. Huh?
That is
synthetic, absolutely synthetic.
So synthetic uncouplers are just molecules, not proteins?
Yeah, they're not proteins. If they were proteins, how would you get them into mitochondria? That would be a nightmare. Okay, getting a protein into mitochondria, oh, you don't want to do that. Very last thing, and I'm sorry I can't spend more time with that. If you want to have more on that, watch last year's lecture. There's quite, I think there's a bit more than that. And because some of you during the break, you asked about what is this energy and how the energy, and if ATP contains more energy than ADP. Okay, this is really important. ATP is what is called, usually in textbooks, is called a macroergic compound. Have you heard macroergic? No? Okay. Macroergic. Okay, macroergic. Comes from Greek and it means lots of work. Okay, it can produce lots of work. Macroergic. So ATP can produce a lot of work. Now, most textbooks get completely wrong the reason why it can cause so much work, and they tell you that there's some special bond which is called a macroergic bond between the phosphorus and whatever. Going back to our thermodynamics lecture, what is the thing that determines how much work we can get from a system? Yeah?
How far away from the equilibrium?
Very good, okay? So either we can say there's the difference in Gibbs energy, which is another way of saying how far it is from the equilibrium. The only thing that determines how much work we can get from a reaction from a system is how far it is from the equilibrium. So ATP doesn't in any way have more energy than ADP. Only our cells keep it so far from the equilibrium, the ATP-ADP ratio, that we can use this difference to produce work, to make work. Okay? If our cells didn't keep it so far away by this whole process, if they didn't keep it so far away from the equilibrium, ATP, even if we had 100 billion tons of ATP, if it was an equilibrium mixture of ATP and ADP, we couldn't produce anything from it. It would just sit there. It wouldn't do anything. It wouldn't power our muscle contraction. It wouldn't do anything. So what makes ATP a macroergic compound, so a useful source of work, is that our cells constantly work to keep the ratio of ATP to ADP very, very far away from equilibrium. In fact, in our cells, the ratio of ATP to ADP is approximately 1,000 to 1. So 1,000 times more molecules of ATP to ADP. The equilibrium mixture of ATP to ADP, so if you took ATP and had it hydrolyzed to ADP as long as it wanted until it reached the equilibrium, is 1 molecule of ATP to 10 million molecules of ADP. So 1 to 10 million is the equilibrium mixture, and our cells have 1,000 to 1. Okay? Okay? About 10 orders of magnitude difference. I think I may have lost you now with the equilibria and... Yes. Okay. Don't have time. But I think last year I had more time to explain it. So if you just watch the very last bit of last year's lecture, you should be able to get it. If you don't, either ask a question on Viuca or come and see me and I'll explain it in more detail. Okay? All right.